Every dot on this plot is a protein structure generated by Rosetta, 366 of them, 4 membrane protein targets. The y-axis is Rosetta total score, the energy. The lower it is, the better the structure is supposed to be. The x-axis is RMSD, how far the structure is from the real protein. Lower means closer to reality. If Rosetta scoring worked reliably, we would see a funnel. The bottom left corner would be dense, good energy, close to native. But there is no funnel. This is the first video of Overfitted, and it's about why. It's 2026, something has shifted in protein design that most people outside the field haven't noticed. Generating protein structures got cheap. AlphaFold 3, released last year, models entire biomolecular complexes, protein with DNA, RNA, ligands, ions, all in a unifying framework. RF Diffusion 3, released by the Institute for Protein Design at the end of 2025, generates proteins that interact with arbitrary cellular molecules. Open source, anyone can run it. If you have a GPU and a few days, you get thousands of candidate structures. But generating structures is only half of the problem. You then have to decide which ones are worth anything. Every protein design workflow looks like this: generate, score, select what to test, test it, learn from what failed. The first step got cheap. The second step is where the field stumbles. Quick about me: I'm Pavel, biochemistry undergrad at Bristol, now finishing my masters in data science and AI in Queen Mary University of London. My dissertation is exactly this question: can machine learning evaluate protein design's outputs better than Rosetta does on its own? The plot you saw at the start isn't from a paper, it's from my analysis of 3 public protein decoy datasets, assembled, parsed, and pulled together for my dissertation. What's open in my notebook right now. Here's what a protein design workflow actually looks like in numbers. You generate 10,000 candidate structures. You score them somehow. You pick 100 for further filtering. Maybe 5 of those work experimentally, and that's a good day. The bottleneck is not the first step. Any generative model can give you structures now. The bottleneck is a second step. Ranking, picking which ones deserve further attention. When you rank structures by one score, energy, sequence, likelihood, predicted confidence, anything, you are making a strong assumption that this metric is monotonically related to a real biological quality. And very often it just isn't. Plausible doesn't mean stable. Stable doesn't mean functional. Functional in a simulation doesn't mean functional in a lab. And working on a benchmark doesn't mean working on the actual protein you care about. Here's the contrast. On the left, what ranking should look like if the score works. Points cluster in the bottom left, low energy, close to native. On the right, what I actually got. Diffuse, mostly horizontal. The score doesn't pick out near-native structures. It just sorts structures by energy, regardless of how close they are to reality. I want to show you the actual numbers. This isn't a literature review. This is from a Jupyter notebook open on my laptop right now. I'm working with 3 datasets, 74 proteins in total, 6,584 structures. The first 3D robot, 58 soluble proteins, almost 6,000 decoys, the standard training pool for this kind of task. The second, Zenodo, 12 small proteins, 360 structures used for independent validation. The third, MPF19, 4 membrane protein targets, 366 decoys. Each one has 27 pre-computed energy terms from Franklin 2019, and energy function specifically calibrated for membrane environments. This is where the things get interesting. Here's the RMSD distribution across all 3. The black dashed line is the 4 Ångströms threshold, the convention for calling a structure near native. Look at the cumulative panel at the bottom left. The red curve is MPF19. For the soluble dataset, about a third of decoys are within 4 Ångströms of the real structure. For the membrane targets, 0. Not one. Out of 366 structures, 0 are within 4 angstroms of native. That alone is important. It means the standard formulation of this problem, rank decoys by quality, find the near natives ones, literally doesn't apply for membrane proteins on this dataset. The pipeline only gives you mediocre answers. Your task isn't picking the great one, it's picking the least bad one. And here's what scoring looks like on this set. Total score against RMSD. One target, V8P, the sign cluster, shows something funnel shaped in the bottom left. The other 3 are diffuse clouds. Total score ranges from -615 to +285. RMSD only from 4 to 13 angstroms. If you sort by total score and take the top 10, you don't get structures closest to native. You get structures within the lowest energy, which can be pretty much anything. Here's the underlying issue. Soluble proteins live in water. Their physics is straightforward. Hydrophobic stuff packs in the middle, polar stuff faces the solvent. The PDB has hundreds of thousands of them. Standard Rosetta energy functions are calibrated on this physics. Membrane proteins live in a lipid bilayer. Their physics is essentially inverted. Hydrophobic on the outside, where the lipids are, polar pocket on the inside. The PDB has maybe 3% of structures from this class. Less data, different physics, different energy functions, different assumptions. And you can see it in the features themselves. Here's the correlation matrix of energy terms in Franklin 2019. Look at FA ATR against FA Sol. Leonard-Jones attractions against the Lazardi-Skarplus salvation term. The correlation is -0.99. They are almost perfectly anti-correlated. Now, to be honest about this, the direction of that correlation is known physics. The original REF15 paper from 2017 explains why. As atoms approach native packing, attractions becomes more favorable, and polar residues get buried, making salvation less favorable. The physics is documented. What's not obvious is how strong it gets on a specific dataset. On mine, the 2 terms carry almost identical information. For a machine learning model, that's the point. You can't just stack these as independent features. The naive feature engineering doesn't work. And that's the largest question that interests me. When a model learns on one kind of data, soluble proteins, and you apply it to a different kind, membrane proteins, what transfers and what breaks? This is a generalization question. It's the heart of machine learning. The channel is called Overfitted, and that's not accidental. Most of scientific ML is a story about a model that works beautifully on its benchmark and breaks on the real problem the benchmark doesn't resemble. Soluble training, membrane application, it's a microcosm of the same thing. If anything matters to me here, it's the evaluation question. How do we know a model has actually learned something, instead of just memorizing its training set? I'm going to document my master's dissertation here as it moves. The next video probably will be about Rosetta. What is it, why it's still used in the era of AlphaFold, and where its energy is informative versus where it lies. That should be up in about, hopefully, 2 weeks. If you work in protein design, computational biology, or machine learning evaluation in general, leave a comment or message me. I'm trying to map this space as I go. Specifically, which evaluation problem in your field is the most underrated? Where does your metric not predict what it's supposed to? That's a real question, not rhetorical. Thanks for watching.